Sea fans and other corals are keystone species that shape the very biodiverse coral reef ecosystems. These corals are threatened by human activity as they are the victims of rising ocean temperatures and habitat destruction. But in addition to these threats, infection and disease can also decimate coral populations. Certain corals are more vulnerable to pathogenic diseases and become even more susceptible due to environmental changes. One such infection is caused by the Aspergillus sedowi fungi, which attacks sea fans. Sea fans and corals have the ability to produce antifungal molecules, but a much stronger antifungal defense mechanism is needed for sea fans, or Gorgonia ventolina, to survive a widespread pathogenic fungal attack. We are the Hopkins iGEM team, and our project this year was on a method to provide this stronger antifungal defense mechanism for sea fans by engineering a symbiotic bacteria. Environmentalism was an important aspect of our project. We recognized that coral health is threatened by human activity, whether it be habitat destruction or rising ocean temperatures. But as a side effect of these ecosystem changes, species of coral become vulnerable to pathogenic diseases. Elevated temperature and nutrition pollution are among a suite of stressors that can contribute to coral mortality from disease. Elevated temperature, which alters the host-pathogen interaction, is widely understood as a key driver of coral disease outbreaks. For sea fans in particular, rising temperatures limit the amount of energy obtained by the sea fan by inhibiting photosynthesis in their symbiont. This makes the corals especially vulnerable to pathogenic diseases, such as Aspergillus sedowi, which typically inhabits terrestrial soils and is hypothesized to have originally reached sea fan colonies due to offshore deposition of terrestrial sediment. Similar to the case of the sea fan and Aspergillus fungi, coral populations in the Caribbean are often impacted by a combination of human activities that weaken their immune response and pathogenic disease. The two main goals of our project are to design a custom genetic circuit for the production of antifungals in response to pathogen presence, and a bacterial containment circuit. The goal of our antifungal circuit is to engineer the cutaneous symbiotic bacterium, Endozychomonas unicicola, that inhabit Gorgonia ventolina to detect and respond effectively to the presence of Aspergillus sedowi. For our containment circuit, our goal was to develop a method where the population of our bacteria was regulated using quorum sensing. We constructed a model to observe the potential decrease in bacteria over time for our designed containment circuit.
In order to reliably detect the presence of an infection, we decided to utilize the sterile regulatory element binding protein, or SREBP, UPC2, in conjunction with a constitutive promoter followed by an SRE. The importance of ergosterol is that it is only found in fungal membranes. The concentration of ergosterol is an important readout of fungal population. UPC2 is inducible by ergosterol. When UPC2 is no longer bound to the SRE, a constitutive promoter can drive transcription. We had several options to choose from for direct killing of the fungi. These included PAF and PAF-B, or Chi-B. We later learned that there may be symbiotic relationships between non-pathogenic fungi and the corals, so we realized that we had to select antifungals with high specificity to Aspergillus sedowi. Chi-B non-specifically targets the chitin in the cell walls of the fungi, so it was a very poor fit. PAF and PAF-B, on the other hand, are far more specific to Aspergillus sedowi and were selected for expression. We realized from prior research on sea fan aspergillosis by Toledo Hernandez and others that the concentration of ergosterol in sea fan tissue decreases upon infection. As a result, we decided to invert the signal such that protein expression occurs in response to a decrease in ergosterol concentration. In order to get antifungals secreted from the cell, we opted for a type 1 secretion system. The system we selected is the highly characterized alpha hemolysin system, which constructs a selective pore that spans both membranes with HLYB, HLYD, and TALC. Any protein, and in this case our antifungals, tagged with HLYA, which will be secreted from the cell. Our team implements two methods to ensure that our engineered bacteria do not invasively spread in the aquatic environment. First, we designed the genetic containment circuit, which utilizes quorum sensing to induce death in cells that drift away from the coral. And secondly, we use agarose hydrogel beads to contain our engineered bacteria upon delivery to the sea fans. Overall, our system is a monostable circuit with dual repression, so the ground state, areas of low bacterial population, entail immediate death, and active AHL is necessary to meet survivable, low toxin conditions. This was inspired by previous work from Shan and others in 2015. Visualized here is our design for toxin synthesis and direct regulation. Our containment circuit involves the production of MASF toxin. The production of MASF is regulated by two repressors. How, you may ask? Well, the TET-R promoter is used to drive toxin expression, so production of TET-R will inhibit toxin expression. The p-aza-R promoter is used to drive TET-R expression, so production of aza-R will inhibit TET-R expression. This constitutes a basic monostable switch with a survival and death state. Since expression of TET-R is quite weak, we will constantly need to induce aza-R to overproduce TET-R and keep the cell in the survival state. Otherwise, it will default to the death state. And how do we induce aza-R? This is through the production of an AHL molecule by the enzyme aza-I. This de-represses TET-R production, which in turn attenuates toxin production. We were concerned that leaky expression of MASF toxin would impact cell survival. As such, we opted for very weak expression of the antitoxin MAS-E to prevent leaky expression of MASF while in the survival state from causing toxicity.
For wet lab, we decided to build the promoter characterization and antifungal production circuits as described before. Here's the general workflow we took to accomplish that. We first attempted 3A assembly of the construct and screened colonies with RFP expression, but assembly ultimately failed several times at the later stages. So we decided to switch over to Gibson assembly, which involved colony PCR and screening with RFP to select for successful colonies, and then we characterized our promoter. This is the 3A assembly schematic. Most of our parts were resuspended and then mini-prepped from the distribution kit. 2 parts, the custom R0010 SRE promoter and the UPC2 coding sequence, needed to be ordered for synthesis from IDT. We used the 3 antibiotics we had on hand, which were ampicillin, kanamycin, and chloramphenicol. We cloned into backbones that carried the RFP coding sequence so that we could screen our colonies. Because of the restriction sites used by BioBrick, in our digestion ligation, the parts that we would insert would replace the RFP. If assembly failed, then RFP would remain in the backbone, and the colonies would express RFP and become visibly red. If assembly succeeded, they would be white. Think of blue-white screening, but with RFP instead. Unfortunately, BioBrick assembly continually failed at the later stages. We decided to do Gibson assembly instead, in hopes that it would get the assembly done more efficiently than 3A. At the first Gibson attempt, we failed the initial PCR to get homologous overhangs on many of our fragments. This gel on the left was the first PCR we did, and the one on the right was a temperature gradient we tried to see what annealing temperature might work best for our primers. We mistakenly designed our primers to have a low TM of around 50 degrees Celsius, so we assumed that might have caused a problem. At the second Gibson attempt, after ordering primers with a higher minimum TM, PCR of most of our parts worked. The only ones that failed were UPC2 and B0015. So we performed a temperature gradient for the 2 parts that failed, and we also tried a different template for uplifting UPC2 from, which was a synthesis we had ordered from IDT, and both were successful. And finally, after performing the assembly reaction, we transformed and plated our colonies, and once again used RFP screening. Next, we characterized our promoter. So our construct contained a circuit where the SRE binding site and GFP coding sequence were downstream of the promoter. UPC2 that was expressed should be bound to the SRE site and be inactivating the promoter. When ergosterol is added, UPC2 should bind to it and then release the site, meaning that ergosterol should induce the promoter. At the same time, high concentrations of ergosterol are cytotoxic, so there should be some optimal level of ergosterol that induce the promoter the most. Promoter activity could be measured by the expression of GFP. So, after incubation with ergosterol for 4 hours, we fixed the bacteria using paraformaldehyde and took plate reader measurements.
After incubation with ergosterol for 4 hours, we fixed the bacteria using PFA and took plate reader measurements to quantify expression. We tested several concentrations of ergosterol and found the optimal concentration for a promoter to be around 0.1 micromolar, which corresponds with literature values for what would be expected during an actual infection. We took this as evidence that the circuit was functional, but our sequencing results showed otherwise. Displayed is our sequence alignment. Notably, the segment containing GFP did not appear at all, so we are honestly puzzled as to why our results showed an increase in fluorescence in the first place.
In addition to our wet lab experience, we also improved a microbial bioreactor developed by the Open Bioeconomy Lab. Our design utilizes a PCB and many custom components to increase the reliability, enhance the functionality, and decrease the size of the prior electrical design. We also designed and tested a custom enclosure to fit our continuous measurement pH probe, as well as a new peristaltic pump for wider tubing sizes, increased flow, and printability without supports.
Displayed are an electrical schematic of all the components we used in our design, as well as the layout of the PCB. This allows us to avoid using jumper wires and breadboards to connect components, and helps our bioreactor to be a more permanent and reliable device. All of the custom parts we designed were printed either on a Prusa i3 Mark III's Plus or a Voron 2.4. These parts had very tight tolerances and fit well, such that the printed lid fits snugly on our culture vessel, and the spindle and our peristaltic pumps fit well without any play onto the shaft of a NEMA 17 stepper motor.
Coral health is incredibly important to many stakeholders, whether they be in government, academia, or coastal communities. Many coral species endemic to the Caribbean are impacted by pathogenic diseases as well as human activity, and solutions are needed to both. Since corals are an important keystone species, their loss would negatively impact the biodiversity of coastal ecosystems, which would have subsequent impacts on other industries, including fishing and tourism. To understand where there are existing threats in coral health and determine how our project could best be implemented, we identified and reached out to stakeholders who provided us with insight into the current state of coral reef conservation efforts. We contacted the Florida Department of Environmental Protection Coral Reef Conservation Program, which works to coordinate several different coral conservation projects. We met with several researchers involved in this program who discussed their work with us. We first met with Dr. Victoria Barker, who is affiliated with the NOAA Coral Reef Conservation Program. We learned from our discussion with her that the current focus of coral conservation research is stony coral tissue loss disease, or SCTLD, which has an unknown cause. This disease attacks stony corals, which provide the structural foundation of coral reefs, and decimates coral populations it is introduced to. We were directed by Dr. Barker towards researchers studying SCTLD treatment delivery methods, which included hydrogels. We then met with Dr. Valerie Paul and Dr. Blake Ushijima, who were both studying methods of treating SCTLD. We discussed what advancement in delivery methods would be helpful with them, and learned that our proposed genetic containment circuit could potentially be helpful. We also met with Dr. Andrew Blake, a coral biologist at University of Miami. We discussed current regulations preventing the release of genetically engineered organisms into open systems with Dr. Blake, and learned that even in unregulated closed systems, such as aquariums, introducing a genetically engineered solution might cause it to be introduced into the ecosystem, as aquariums are often disposed of in this way. We collaborated with several iGEM teams over the course of the year. Our human practices team collaborated with Florida State University iGEM. Since our project was on a subject of interest in Florida, we got advice from the FSU iGEM team that helped us develop our stakeholder list and identify groups we could reach out to. We also helped them get feedback on human practices aspects of their project, which focused on food insecurity. Our human practices team also worked with MIT iGEM to develop an educational module on molecular cloning. Our dry lab and wet lab teams both worked with University of Florida iGEM. Our dry lab team discussed the design process and methods of designing a logo and wiki with UF iGEM, while their wet lab team reviewed our assembly plan for the antifungal production circuit and provided us with a custom hi-fi assembly protocol. Our wet lab team also provided some mentorship to the Baltimore BioCrew iGEM team. To coordinate these collaborations, several of these iGEM teams formed the Eastern Seaboard Group. This group met occasionally to discuss project progress and identify where each of the individual groups could benefit from the collaboration. This group helped with identifying beneficial collaborations and keeping all the teams up to date on each other's progress. Several of these teams also worked on a collaborative Minecraft server to showcase our work. Our educational module was made in collaboration with MIT. Our teams noticed that there is a lack of educational materials that make learning about molecular cloning techniques more accessible. And our goal with this module was to create an easy-to-understand educational module that inspires people to learn more about synthetic biology. We produced a video on type 2S restriction enzymes for this collaboration.
For the dry lab, we intend to proceed by refining our current containment model by minimizing the effects of the previously mentioned limitations. A more involved diffusion simulation is currently in the works, and once diffusion can be modeled reasonably well, we intend to rerun tests by populating the model with various numbers of cells to see if the containment circuit works among various numbers of cells as opposed to only within a single cell. We'd also like to model the ergosterol pathway within the target fungi in order to verify the antifungal agent works as expected. Once these two pathways are fully modeled, we could then proceed to model the pathways together in order to visualize the interactions of the pathogen with our bacteria, as well as potentially the coral's immune system to either of these organisms.
For wet lab, our next step is to quantify the antifungal production ability of our E. coli to see if PAF and PAF-B will be expressed at meaningful quantities against fungal pathogens. Second, we would build a genetic containment circuit and test whether it works in vivo, and whether bacteria experience cell death at low populations. And we would also test whether they survive when coated in hydrogel beads. Finally, we would co-culture the bacteria with A. sedawi and see to what extent they are able to kill the pathogen or restrict its growth.
For our human practices team, the next step would be to further our collaborations with coral research groups. The team has members who are with us for the duration of this project, as well as new members who joined as trainees this semester. We would also like to thank our faculty advisors, Dr. Winston Tim and Dr. Jessica Dunleavy, as well as our student advisors, Ethan Levy and Angel Lee. We would also like to thank the JHU Department of Biomedical Engineering, Agara Bio, IDT, and NEB